Kimitec Group is a company leader in crop bionutrition and bioprotection. We are present in more than 90 different countries with a unique philosophy of the application of natural resources against pests or different crop needs. In a world dominated for multinational companies that offer to the market chemical solutions, Kimitec Group, thanks to the BLOSTER Project, has been consolidated as a natural, sustainable, homogeneous and high-efficacy solution against the chemical treatments, with a model based on investigation, innovation, nature, and finally common benefit for all of us.
The following video shows us using our scientific method with an optimized technological process throughout the execution of the project. This ranges from the natural origin of raw materials to the final formulation of the product, using different biotechnological processes for obtaining the active ingredients. The BLOSTER Project has aimed to design and produce a new generation of biopesticides comprised of 5 specific formulations for controlling pest diseases of great global impact, such as whitefly, trips, nematodes, aphids, and botrytis. The unique formulation technology developed in the BLOSTER Project integrates in the final product a botanical component from terrestrial and marine plants, metabolites of microbiological origin, defense inducers, and co-formulants of natural origins. The final result translates into a product with great versatile action mechanisms, efficiency, and homogeneity, equalizing and overcoming the effectiveness of conventional synthesis products. The good performance of the BLOSTER Project depends on a previous analysis that allows us to detect and perceive the need of each potential client, followed by an in-depth market study to outline the project strategy with each of the products, including the identification of the pest or nutritional problems in a given target crop and within a specific country. The final success of the formula is in the composition and combination of its active ingredients. Kimitec has applied within BLOSTER a unique methodology of searching with detailed study of raw materials based on the design of specific documentary blocks for each of the pests and target diseases. This permits the selection and prioritization of raw materials based on the valorization of agri-food waste and the cultivation of domesticated endemic plants. Discarded bulbs have been used during the cultivation and processing of medicinal condimentary spices, also waste generated from the wine industry. Another source of raw materials has been the cultivation of endemic plant chemotypes, whose satisfactory adaptation to the conditions of southeast Spain have been one of the main milestones reached in the project, and a source of our own homogeneous raw materials for the development of our biopesticides. Our line of production and optimization of formulations include the design, development, and optimization of extraction, separation processes of active principles from selected raw materials. The collected plant material undergoes a specific drying and micronization to guarantee the success of the extraction process. The extraction technology will depend on the raw material selected and include from conventional methods of steam distillation to more advanced methods that use multifunctional dispersion systems and fermentation biotechnological processes. Each of the variables of the selected process are studied and optimized in pilot prototypes on a laboratory level, and subsequently we produce a scaling process to an industrial level. At the end of this process, the crude extracts are obtained, which proceed to the characterization phase. After each extraction process, the different batches obtained are characterized based on a metabolomic point of view and biological activity to test their homogeneity, stability, and effectiveness. The metabolomic profile is determined by the chromatographic and spectroscopic methods as well as chemometric analysis that provides relevant information on the exact chemical composition of the extract. The biocidal activity is determined through microtests at the laboratory plant field level, applying specific biomodels designed and adapted from the specialized literature of Kimitec for each of the pests and target diseases included in the project. During this stage, the effective doses are determined, the different action mechanisms are discriminated, and the possible structure-activity relationship are established. Each formulation is elaborated from the application of a formulation technology and design of specific synergies depending on the chemical composition and mode of action proposed. It includes controlled release systems, nano and micro encapsulations, adhesion biofilms, highly stable microemulsions, colloid mill, and thixotropic dispersion mechanism. At the same time, stability, rheological properties, and physical chemical characteristics studied of the formulations are carried out, while also the design of their labeling and packaging. The final products are sent to accredited laboratories to carry out the toxicological and ecotoxicological tests required by the countries selected for their registration. Each of the designed biopesticides are subject to validation tests for their biological efficiency under semi-field, greenhouse, and open field conditions at the effective doses recommended in the previous laboratory tests. These tests are carried out according to the statistical experimental designs and include dose studies, phytotoxicity, safety periods, knockdown effects, permanence, systemic characteristics, effects on auxiliary fauna, among many others. With all the information gathered during the documentation, processing of raw materials, production process, formulation, chemical composition, laboratory trials, field trials, and toxicological trials, a dossier is prepared to present in registration procedures. In addition, all our formulations carry a life cycle analysis based on the environmental footprint methodology established by the European Union. As a result of the BLOSTER project, 5 formulations have been obtained to control whitefly, trips, nematodes, aphids, and botrytis. The bioproducts obtained have great versatility of action mechanisms, efficiency, and homogeneity that equal or surpass the effectiveness of conventional synthesis products, with the advantage of directly attacking the problem related to the development of cross-resistance, while promoting a green economy and helping us return to a more sustainable agriculture. This project has been co-financed by the SME instrument of the Executive Agency for Small and Medium Enterprises within the Horizon 2020 program of the European Commission.